Good afternoon. Today we are going to see the split genes. So, what are split genes? Usually it is considered that a gene is contagious, means when the gene undergoes a transcription process, usually the nucleotide sequence which is present in the DNA is transcribed into a messenger RNA molecule. So, the corresponding nucleotide of the DNA and the nucleotide of the messenger RNA matches, means there is a one to one correspondence in them. But in the split genes, what happens? If you observe the messenger RNA sequence, the nucleotide sequence doesn't match or doesn't match one to one with the nucleotide sequence of the DNA. It means that few portion or some portion of the messenger RNA is being cut, and this is because that the messenger RNA is formed only from the coding sequences and the non-coding sequences are not transcribed into a functional messenger RNA. So, that means that whatever the removed genes are present, that removed genes fragments of the nucleotide sequence, they are called as the introns or the intervening sequences. So, in split genes we will see that split genes, when they are have a interrupted sequence of nucleotides, then they are called as a split genes. So, as I have said, if there is no one to one correspondence and if the genetic map is not corresponding, means the genetic map of the messenger RNA is not having the same length as that of the DNA, then you can say that it consists of the split genes. So, usually genes has a continuous sequence of nucleotides. In other words, there is no interruption in the nucleotide sequence of a gene, and therefore the nucleotide sequence as a whole, it undergoes a transcription process and then a translation process to form a particular single polypeptide chain. So, but however, you will see that a sequence of nucleotide which is not contained in some genes, the sequences of nucleotides were interrupted by these intervening sequences, or these intervening sequences which have not got transcribed into a polypeptide molecule. So, how this discovery was made? The split genes were independently discovered by Richards J. Roberts and Philip A. Sharp in 1977, for which they shared Nobel Prize in Physiology and Medicine in 1993. So, the first observation of interrupted genes, that is the genes in which there are non-coding intron sequences between the coding exons sequences, were made in animal viruses in 1977. This is about the history of split genes, how the split genes were discovered. So, split genes were discovered by Richards J. Roberts and Sharp in 1977, and then later on intron sequences or the intervening sequences in animal viruses were observed in 1977 too. So, what does it mean? The split genes, they have two types of sequences. One is called as the normal sequence, which you refer is as the exons, and the another one is called the interrupted sequence, or you call it as the introns. So, now what are the normal sequences? Normal sequence means the sequence of nucleotides which are included in the messenger RNA, which is included in the messenger RNA and is translated from the DNA of a split gene. So, this the nucleotide sequence which is present in the DNA, and from the DNA you get a messenger RNA and then you get a particular polypeptide chain. So, this is what is called as the exons or the normal sequences. But what do you mean by the okay? So, by this diagram you will observe that here you have the intervening sequence or the introns. So, and this is your coding region or the exons. So from coding regions or the exons only you will get a messenger RNA. But these two intervening sequences which are present, this is one intervening sequence which is non-coding region, and this is your second intervening sequence which is a non-coding. So this will not get transcribed into the mature messenger RNA. So what happens when it undergoes a transcription process? Usually the entire DNA undergoes a transcription process, but these introns get loop out. So you observe this diagram, you will see that the introns are getting looping out. So this is your pre-transcript. Pre-transcript means which contains the region of the introns as well as the exons. Now after that the splicing of the introns takes place. So this region is cut off, and this region of exon is joined together with the second region of the exon, and this intron is eliminated out. Then the second region of exon is joined with the third region of exon, and this second intron or intervening sequence is removed out. So when the splicing is done, usually you get a messenger RNA. So this is how it is taking place in eukaryotic cell. So in the nucleus you have a DNA, and in nucleus itself you will see that the transcription process takes place. So when transcription is taking place, you will find a transcript which is made up of the exons as well as the introns. And then what happens, this is referred as the pre-transcript. When you get a messenger RNA, this is what is referred as the pre-transcript. So this intron region are shown in red region, and the exon regions, they are shown in the yellow region. So what you have a 5' capping and a 3' poly-A tail, because when it is coming out into the cytoplasm for protein synthesis, usually it has to undergo a modification like splicing of the introns, then 5' capping and 3' poly-A tail addition. So usually this is done, and then you get a messenger RNA, and this mature messenger RNA now enters through the nuclear pore and comes out into the that is, it enters through and comes out into the cytoplasm for protein synthesis. Okay. So these are the intervening sequences. These intervening sequences are usually called as the introns, and these do not code for any peptide molecule, and therefore they are spliced out or they are removed out. So that is what is called as the split genes. Now each interrupted gene begins with an exon and also it ends with an exon. If you observe the gene, you will see that it starts with the exon and it ends with the exon. Understood? So it is always starting with the exon and it is ending with the exon, but in the middle you have the introns. Then the exons occur in the same precise order in the messenger RNA in which they occur in the gene. So exons are usually the same, the nucleotide sequence present in the exons, they are usually occurring as it is in the DNA, that is the coding strand of DNA and the messenger RNA strand. The same interrupted gene organization is consistently presented in all the tissues of the organism. So you will observe that the introns are not representing in the messenger RNA, it is only the exon which is represented in the messenger RNA. Now most introns are blocked in all reading frames because the termination codons occur frequently in their 3 reading frames, and therefore most introns do not seem to have coding functions. Because of this, they do not have the coding function. Now we will see the significance of split genes. The significance of split organization of eukaryotic genes is not clear, but in some cases different exons of gene code for different active regions of the protein molecules. For example, in the case of antibodies, it has been suggested that introns are relics of evolutionary processes that brought together different ancestral genes to form a new larger genes, and it is possible that some introns have been introduced within certain exons during the evolution. So, this proves that there is some evolutionary relationship in between the genes, so that during the formation of antibodies, usually whatever the genes were brought from the ancestral ones, now they are located in the daughters or in the progeny cell, so that this has developed over a period of time, and therefore you will see that you have got a evolution, or you have there is a change in the genes, and these genes have now become useless or non-functional, or you call it as the redundant genes. So, introns may also provide for increased recombinational rates between the exons of the gene. So, they also provide the recombinational rates between exons of the genes, and thus for few cases you will have a significance in genetic variations. Because of the presence of introns, you will see that there is increased recombination rate, and therefore you will see variation in the genetic material. Introns are known to code for enzymes involved in processing of heteronucleus or heterogeneous RNA. So, usually these introns, they also code for the enzyme which is used for processing the heterogeneous or heteronucleus RNA molecule. So, we have altogether seen. Now, we will see the examples one by one. So, it is generally believed that a gene is a contiguous or in continuous, because it is not having any uninterrupted sequence, but in split genes you have uninterrupted sequence, which you call it as a intron. So, the recent work shows that the sequence of eukaryotic genes like globin and ovalbumin is present in the oviduct of hen, which is responsible for synthesis of ovalbumin protein. So, and the globin gene, beta globin gene, which is present in mouse, that is in the hemoglobin. So, hemoglobin is containing the globin protein part, and this hemoglobin is usually is having the interrupted sequence or the introns. So, although the sequence of the sequence are transcribed into heteronuclear or the heterogeneous RNA, they are later on spliced together, excised, the introns are spliced, and the exons are joined to make a mature messenger RNA, and then it is translated into a protein molecule or the enzyme molecule. So, such interruptions within the sequence of the gene have been called as introns, or they are referred as inserts or intervening sequence, or it is referred as silent DNA. So, the sequence are included in the messenger RNA and translated, which have been called as the exons. Already I have explained you what is introns and what is exons. Exons are those coding regions where they these regions, which is a nucleotide sequence on the DNA, which is getting transcribed to form a messenger RNA. So, eukaryotic genes, usually it is a mosaic of introns and exons. So, you will observe that the coding regions, they are interrupted with the non-coding one, or non-coding one is also called as a spacer, but better it is you refer it with the introns. So, hence it is called as the split genes. Now, if this is your DNA, then EG is the exon 1, this is your intron, or you call it as a spacer, then this is your second gene EG2, then you have again a spacer, then here you have a gene 3 exon. Okay. So, this is the region where it is the expressed genes which you observe after transcription, which undergoes a translation process to give you a functional protein molecule. and again you have a spacer or the intron and then express gene 4. So, when it is undergoing transcription, the complete sequence of DNA is corresponding with the RNA, but this RNA is called as a heterogeneous RNA. Okay. So, here this portion is transcribed, but it will then get excised out or removed out. So, this region is removed out, this region is removed out, and this region is removed out, and then the splicing of this only the exons takes place, that is the express gene G1, G2, G3 and G4, and then you will get a much smaller messenger RNA which is finally translated. It means that if you see the length of this messenger RNA and if you observe the nucleotide sequence of this mature messenger RNA, this is what is called as a mature messenger RNA, and if you try to correspond it with the nucleotide sequence of DNA, then obviously you will see that the DNA nucleotide sequence is much larger than the messenger RNA. Why it is so? It is so because these introns regions are not coded and therefore they are not included in the messenger RNA. Okay. So, it is called as the that particular introns, or this is called as the split genes, because the gene is being splitted. Where it is being splitted? It is splitted at the region of non-coding or at the region of intervening sequences. So, these intervening sequences were unequivocally demonstrated for first time in the gene of beta globin in mouse and rabbit, and then in chick ovalbumin. So, in the beta globin gene of mouse and rabbit, a single intervening sequence was present, and this intervening sequence is near about 200 to 250 nucleotides inside from one end of the gene in both somatic and germ line cells of the animals. Understood? So, this beta globin gene of mouse and rabbit is intervening sequence which is only a single intervening sequence about 200 to 250 nucleotides and is present in both somatic and germ line cells. Okay. So, how they demonstrated it? The White and Hognes, these two scientists, in 1977, they developed a R-loop mapping technique for identifying the site and length of intervening sequence in the electron microscope. So, the method involves, how this method is done? This method involves hybridization of RNA transcript, means the transcript you get or you obtain after transcription process with double helical DNA of the gene at high temperature and increased concentration of formamide. So, when it was subjected to higher temperature and increased concentration of formamide, under these conditions what happens? RNA binds with the DNA hybrid molecules to form a stable, more stable than DNA duplex. DNA duplex means DNA and RNA DNA, it forms a hybrid. So, when they observed in those regions where RNA is complementary to the DNA strand, the two form a hybrid duplex, and wherever there is no complementarity, that unpaired DNA strand is forming a single stranded loop which is visible in the electron microscope. This is explained better in the diagram. See, if you observe, this is your mature messenger RNA, and when it was usually hybridized, this is your RNA DNA hybrid. When it was hybridized at increased temperature and higher concentration of formamide, you will observe that where there is a complementarity, the DNA has got DNA RNA has formed a hybrid structure, but this region is where it has got looped out, since the single stranded DNA is not having any complementary nucleotide sequence. So, it means that the DNA length or the whole of or the complete DNA sequence of is not getting transcribed into a mature messenger RNA molecule. Okay. So with this technique, Timur found in 1978 found out that intervening sequence in beta globin gene forms a R loop of about 585 base pairs long, and it is located about 200 to 250 base pairs inside the 3' end. At the 3' end it was located. So the formation of loops were also shown or observed in adenovirus and SV40. So the ovalbumin gene in chicken has been shown to contain 7 intervening. See, in beta globin gene you have a single intervening sequence which is about 200 to 250 base pairs at the 3' end, inside the 3' end. And here usually what happens in the ovalbumin gene, you have 7 intervening sequence. By the combined technique of restriction enzymes and R looping, this particular sequence of matching has been shown in figure, which I will be showing you. So the insulin gene in mammals has 2 intervening sequences. So this is the Hexon gene of adenovirus, as I have already explained, that is A, B, C represents the intervening sequence. So here you will see that this region has been looped out because it is non-coding region or the intron region. This region B and then C is here itself, as well as this is looped out in this fashion. Okay. So this is how you will represent the intervening sequence in the Hexon gene of adenovirus. In mammals, rat has 2 insulin genes, 1 and 2. So the gene for insulin 2 has 2 intervening sequence. One sequence is of 119 base pairs and the other is having 499 base pair longs. So even in chloroplast and mitochondria of eukaryotes, the genes for the larger RNA contains intervening sequences. So as you know that in the Hexon gene or the beta globin gene, you have the intervening sequence. Because of this, you will see that there is a split genes. Okay, so now we'll explain about the ovalbumin gene. So as I have said, there are 7 intervening sequences. So this is your coding region, then S1 is introns or intervening sequence, S2 is another intron, S3, S4, S5, S6, S7. Okay. After formation of a RNA transcript, usually messenger RNA, then what happens, you will get a heterogeneous RNA. Then this heterogeneous RNA is getting looped out. Got it clear? So after transcription and formation of a mature messenger RNA, this mature messenger RNA, if you compare it with this particular DNA and forming a RNA DNA hybrid, this DNA gets looped out at 7 regions because they carry 7 intervening sequences. So this is the structure of ovalbumin gene and looping out of 7 intervening sequences. So EM studies on cloned nucleolar ribosomal RNA genes, they have shown it contains the intervening sequence. In fact, one of the first reports for split gene was on 28S rRNA. So when the rRNA gene is transcribed in nucleolus, in it was first observed in Drosophila melanogaster. So there is a spelling mistake here. So Drosophila melanogaster. So these genes were found to be of variable lengths. So when the complementary rRNA was hybridized with the rDNA, then obviously the rDNA was longer than the rRNA. It means that there are intervening sequences which is not forming the nucleotide sequence of the that is ribosomal RNA. Okay, so they are not represented in the rRNA. The genes for tyrosine and phenylalanine tRNA also shows the short intervening sequences of less than 20 bases, and it was first found out in the East that intervening sequences of less than 20 bases were observed in the phenylalanine and tyrosine tRNA. So, these usually 20 bases in the region of coding for the anticodon loop of tRNA. So, where they were located? They were located in the anticodon loops of tRNA. The intervening sequence of less than 20 bases were located in the coding region of anticodon loop of tRNA. Now, lately split genes have been reported from various materials, and post-transcriptional removal of intervening sequences, they appear to be quite common in many eukaryotic genes. So, introns, exon structures of split genes. So, you have already seen the RNA DNA hybrids and the electron micrographic study or electron microscopic images of these RNA DNA hybrids have shown that the sequence of DNA and complementary DNAs or the cDNA demonstrated only the coding region of mouse beta globin gene. So, why it is called as the beta globin? Because it only codes for the beta subunit of hemoglobin or the globin protein, and it is interrupted by two introns which are spliced out from the messenger RNA. So, in general, intron exon structures of eukaryotic genes is complicated. The amount of DNA in intron sequences usually it exceeds, means it is much longer in size than that in the exons. For example, the chicken Ovalbumin gene contains 8 exons and 7 introns. So, as we have seen, the Ovalbumin gene is having 7 introns and 8 exons over the coding region. They are distributed over 7,700 base pairs, that is 7.7 kilobases of genomic DNA. The exon totals only about 1.9 kb, which means that 75% of the gene is consisting only of introns, means only 25% of the gene is coding for this functional that is RNA, messenger RNA, which is making your ovalbumin protein, and 75% of the gene is only made up of intervening sequence which is of no use. Understood? So, next notable example is the human gene, which encodes the blood clotting factor 8. This gene contains 186 kilobase pair of DNA and is divided into 26 exons. So, you have 26 exons, and these 26 exons carry, if you observe the messenger RNA of this spliced 26 exons, it is only about 9 kb long, which implies that 175 kb of the gene contains the introns. So, from 186 kb of DNA, it is the messenger RNA is only about 9 kb, means you have to minus 9 from 186 kb. So, you will get that is 175 kb. It means that 175 kb of introns or the nucleotides is of no use. Only 9 kb is used. On average, intron constitutes about 10 times more DNA than exons in the genes of higher eukaryotic cells. So, because of this only you will see that even if the human chromosome is having 3.1 billion base pairs, see, remember the human chromosome or the chromosome is having 3.1 billion nucleotide sequence or the base pair. Among those, if you observe the number of genes, there is only 25 to 30,000 genes. It means that in between these are the functional genes, but the other genes means otherwise it should have 5 million genes, but it is having only 25 to 30,000 functional genes. It means that the remaining nucleotide sequence, the remaining nucleotide sequence is only a redundant one which is of no use for the higher eukaryotic organism. Although most eukaryotic genes contain introns, their presence is not universal. Histone genes do contain intron, which shows that introns are not required for gene function in eukaryotic cell. Usually all the genes they do not carry introns, but you find the introns in the eukaryotic cell. Most genes of simple eukaryotic yeast lacks introns. So introns, however, they are present in some rare genes of prokaryotes also. So most introns have no cellular function, although a few have been found to encode functional RNAs or protein molecule, or they can function as enzymes also. In general, introns are considered to represent remnants of the remnant means redundants, which is of no use of sequences that were important earlier in the evolution. For example, I will give you the best example. So when the frog is developing, in the tadpole stage of development of frog, you have a tail like region. Okay, so the genes responsible for having a tail like regions, when the frog is getting matured, that tail like region is of no use. It means that the genes which were of use when it was in the fetal stage, that genes is of no use when the frog matures. So that genes are present on the DNA in the cells of frog, but that is of no use when the frog is getting fully developed. So such genes are also observed in the humans also, because the certain genes, they are required during the fetal growth, but later on that fetal genes, they are of no use when the human beings or the child is growing stage by stage. So these genes are remaining in the cells, but they are of no use. So introns, you will see that some genes, they were related with the evolution process. So how the from chimpanzees, how the humans have got evolved, the genes which were present in the chimpanzees were very related, means 99% correlation was there with the human beings, but during the evolutionary stages, few genes have got redundant and we have got evolved through changes in the genes. So they have accelerated the evolution. So these introns, they are considered, or they have helped to accelerate evolution by facilitating recombination between the exons of different genes, and therefore this process is also called as the exon shuffling. So recombination between introns of different genes could produce new genes containing novel combinations of exons, because when they undergo a recombination, you will observe a totally new exon, and this totally new exon helps in the evolution, and because of that you will see that there is a formation of chimeras. Chimeras means when two different exons are recombining together in different ways, you will have a chimera of the exons derived from several other genes, and because of these new genes, it is so because the recombinational event has taken place in between the intron sequences. Okay. So thank you. I hope you have understood this lecture. Thank you very much.